DNA replication. DNA replication is a biological process of producing two identical copies of DNA from one original DNA molecule. It occurs in living organisms. It acts as most important part of biological inheritance. DNA. DNA is a molecule made up of two strands twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases, represented as A, T, G, and C. Two strands are complementary to each other. A comes with T and G comes with C always. Each strand has 5' and 3' end. Two strands run in opposite direction. DNA replication. At first, two strands are separated by unwinding, and it is done by the enzyme helicase. Helicase enzyme breaks hydrogen bond holding complementary base of DNA together. This results in formation of bubble-shaped replication fork. The separated strand each provide a template for creating new strand of DNA. Primase enzyme starts the process of DNA replication. It makes small pieces of RNA called primer. Primer marks the starting point for the construction of new strand of DNA. DNA polymerase enzymes binds to primer and make a new strand of DNA. DNA polymerase can only add DNA bases in one direction from 5' to 3' end. One of the strand of DNA is called leading strand as it runs from 5' to 3' direction and it is continuous. Another strand of DNA called lagging strand cannot be made continuously because it runs up in opposite direction, that is in 3' to 5' direction. Whereas DNA polymerase enzymes work only in 5' to 3' direction for the formation of DNA bases. In the lagging strand, DNA polymerase make the strand in series of small chunks called Okazaki fragments. Each fragments start with RNA primer. DNA polymerase then adds a short row of DNA bases in 5' to 3' direction of lagging strand. Next primer is added further down the lagging strand. Another Okazaki fragments are made and the process is repeated. Once new DNA is made, exonuclease enzymes removes all the RNA primer from both the strands of DNA. Another DNA polymerase fills the gap left behind the DNA by RNA primer. Finally, DNA ligase enzymes seals up the fragments in both the strands to form continuous double-strand DNA molecule. In this way, DNA replication takes place and multiple copies of DNA are formed. DNA replication is called semi-conservative method because DNA is made up of one old conserved strand and one new strand. Thank you.